Hello and welcome everybody to my channel Lumini Bio Entrance, and today we will be discussing University of Hyderabad PhD Entrance Examination Biotechnology. Even though I have written biology part only, I have discussed some mathematics questions also. As only thing I have not discussed is chemistry questions I have not discussed. Coming to the first question. The length of a ladder that is leaning against a wall making 60-degree angle with the ground and foot of the ladder is resting 4.6 meters away from the wall. So we have to find the length of the ladder that is leaning against the wall. So here, 1 minute. Here x be the ladder, AC be the ladder. So that will x be the length of the ladder. It is making an angle of 60 degree with the wall BC. So B is the foot of the wall, and given angle CAB is the angle that is 60 degrees. So in a right triangle, angle CAB is equal to cos 60 degrees, that is equal to AB by AC. So AB by AC, or AC is equal to AB by cos 60. We know cos 60 degrees is half. So AC is x meters, so that will be equal to AB is given as 4.6 meters. So 4.6 divided by 0.5, that will give you 9.2. So the answer here is option D. Find the missing number. 1 is 1, 28, 92, 217, 433, 776. So here what they have done is they have added 1 plus 3 cube, that is giving you 28. 28 plus 4 cube will give you 92. 92 plus 5 cube will give you 217, and like that 776 plus 8 cube will give you 1288. So the answer here is option C, that is 1288. The increasing order of pKa of the following amino acid in aqueous solution is glycine, aspartic acid, lysine, arginine. So based on the acidic strength, you should know the side groups of these amino acid. Glycine is having only H as the amino acid, so it is neutral. Then aspartic acid has 2 COH group, lysine has got basic group as well as acidic group, and arginine also has got basic group as well as acidic group. And based on the acidic strength, it will be aspartic acid greater than glycine, greater than arginine, greater than lysine. If you, the same thing, if you give in the less than sequence order, that will give you the increasing pKa of the amino acids. So pKa will be aspartic acid, glycine, arginine and lysine. So answer here is option A. Increasing temperature results in. So increasing temperature, it results in hyperchromic shift. Now what is hyperchromic shift? An increase in absorption of UV light by DNA solution due to heat, alkaline conditions, etc. Is called as hyperchromic shift. The shift is caused because of the disruption of the hydrogen bonds of each DNA duplex to yield single-stranded structure. That is called as hyperchromic shift. Hypochromic shift is just the opposite of that. That is, it describes the decrease in the absorbance of UV light in the double-stranded DNA compared to the single-stranded form. Bathochromic shift is an effect due to which the absorption maxima is shifted towards a larger wavelength by a change of the polarity of the solvent. That is called as bathochromic shift. And hypsochromic shift is the change in the spectral band position in the absorption, reflectance, transmittance or emission spectrum of a molecule to a shorter wavelength. So increase in temperature results in hyperchromic shift. Next one. Which of the following agents is used for DNA quantification? It is diphenylamine. That is CTAB and all. It is used in the extraction of DNA from plants, not quantification. Then next one. Clino fragment is derived from. So clino fragment, it is derived from DNA polymerase 1. Answer is option B. The enzyme sequences that is used in DNA sequencing has. So sequences DNA polymerase, it is a genetically engineered form of T7 DNA polymerase. So here option D, that is derived from bacterial DNA polymerase 1, is wrong. It has no 3' 5' exonuclease activity. Then it is appropriate for the dideoxy-mediated chain termination method to determine the sequence of the DNA. So it does not have 5' 3' exonuclease activity, nor it does have 3' 5' polymerization activity. It has high processivity. Answer is option A. Then, if the given amino acid sequence is digested with trypsin enzyme, how many peptides will be released from it? So, trypsin, it cleaves on the C terminal of lysine and arginine residue. So, here you will get one cleavage over here, that is here, then lysine again here, but lysine is followed by proline, so it will not cleave there. Instead, it will cleave the arginine over here. So how many peptides you will get? 1, 2, 3, 4. So the answer here is option C, that is 4 peptides. The next one. In serine protease, nucleophile is activated primarily by. So it is by histidine that the nucleophile is activated in serine proteases. Answer is option B. Which ligand will be employed to purify GST tagged fusion protein by affinity chromatography? So GST is glutathione S transferase. So your answer is there in the question itself. So GST tagged protein, it is purification is based on affinity of GST to glutathione ligand. So the answer here will be option D. The next one. If protein is unable to bind to the matrix during affinity chromatography, what should not be done to troubleshoot? They have asked what should not. To troubleshoot it among the following. Adjust buffer conditions that you can do. Check plasmid sequence. Yes, that also you can do. From plasmid sequence, you will come to know the protein sequence. From there, you can see whether there is any mistake in your protein sequence that it is not binding to the affinity column. Decrease the flow rate of the column. That also is possible. Wash with higher stringency buffer. That is not possible. Answer is option D. In ubiquitination process, which enzyme establishes a thioester bond with ubiquitin? So ubiquitin, it requires 3 enzymes, that is ubiquitin activating enzymes or E1, ubiquitin conjugating enzymes E2 and ubiquitin ligases E3. So here they have asked which enzyme establishes a thioester bond with ubiquitin. So it is E1 or ubiquitin activating enzymes. So the answer here is option A, E1. Then next one. A forms complex with L1 with dissociation constant of K1 and forms complex with L2 with dissociation constant of K2. If K2 is more than K1, which is more stable? So higher the value of dissociation constant, lesser is the stability. So AL1 is more stable than AL2. Answer is option A. Next one. Which of the following is not a segmentation method? So it is dendrogram, that is not a segmentation method. It is in bioinformatics. So answer here is option D. The next one. Rukmini wished to wish her friend Sayani on her recovery from illness. She started from her school and cycled 20 meters towards north. So this is it, 20 meters towards north. N I have written over here to show that it is north. Then she turned right and went 30 meters to a flower shop. That is, she will turn right when 30 meters to a flower shop. After buying flowers, she cycled in the right direction. That is here, this way, for 35 meters to reach the chocolate shop. This is the chocolate shop over here. So 35 meters she cycled to reach the chocolate shop. There she brought some chocolates and started cycling left for 15 minutes. For 15 meters, sorry. So she turned left and cycled for 15 meters. From there she took another left and moved 15 meters to reach Sayani's house. Then she turned left, moved 15 meters to reach Sayani's house. So Sayani's house is over here. In which direction and how many meters is she from her school? So this is the direction and this is the school. So this is the direction from where she is from her school. And how many meters? So how many meters will be 30 plus 15. 30 plus 15 is 45 meters east. So the answer here option A. Then next one. Match the following. So PubMed, AlphaFold, DIP, Interpro. PubMed is it is a resource of protein-protein interactions. PubMed. AlphaFold. Sorry, it is a literature database. Okay, I'm sorry, extremely sorry for that. PubMed is a literature database. Then AlphaFold is predicted protein structure resource. DIP is DIP. It is a resource of protein-protein interaction. And Interpro is it provides functional analysis of proteins. So the answer here is option B. Then next one. Calculate the angle between the hour hand and the minute hand of a clock at 5:30. So at 5:30 the hour hand rests halfway between the 5 and 6 and the minute hand is exactly at 6. All of you know that how it will be at 5:30. As there are 24 hours interval on a clock, the angle of one is 360 divided by 24, that is 15 degree Celsius. As the bands are 1/2 hour interval apart, as the hands are 1/2 hour interval apart, they are 15 degrees apart. So the answer here is option A, that is 15 degrees. Then next one. SCOP, SCOP. SCOP is database for comparing and classifying protein structures. PDB, Protein Data Bank. It is a repository of the 3D structure of biological molecules. PAM is a substitution matrix. String is resource of protein-protein interactions. So the answer here is option B. Then next one, sucrose shows. So for Tollens and Fellings reagents, whether sucrose is positive or negative. So Fellings test, it is based on the principle that presence of aldehydes can be detected easily because of their reducing property. Now this test, it is mainly used for reducing sugars that contain aldehyde group. Now sucrose does not contain a free aldehyde group and is non-reducing sugar, and therefore it will be negative to Fellings reagent. Then Tollens test is shown by reducing sugars that contain free aldehyde group, but sucrose, as I told you already, it is a non-reducing sugar and it doesn't have a free aldehyde group, so it is not going to show Tollens test also. So the answer here will be option B, that is negative with Tollens and Fellings reagents. Then next one, find the probability that a leap year has 52 Sundays. So there are 52 weeks in a year, so there will be 52 Sundays. So 52 into 7, that is 364, but we have 365 days. So that leaves a day for normal year and 2 days for a leap year. So every leap year has 52 Sundays. Now the probability that depends on the remaining 2 days. So let us look at the combination of the 2 days possible. So Sunday, Monday, Monday, Tuesday, Tuesday, Wednesday, Wednesday, Thursday, Thursday, Friday, Friday, Saturday, Saturday, Sunday. So you get Sunday only for 2 days, 2 possible combinations. That is this one and this one. To get exactly 52 Sundays, none of the last 2 days should be a Sunday. That is possible in 5 out of 7 cases. So ignoring these two, you will get 1, 2, 3, 4 and 5. So the possibility, so that is possible in 5 out of 7 cases. So the probability will be 5 by 7. Answer is option C. Then normalization of transcript expression is performed by calculating. So it is performed by calculating what is called as RPKM. RPKM is reads per kilobase of transcript per million mapped reads. It is the normalized unit of transcript expression. So RPKM, that is reads per kilobase of transcript per million mapped reads. Now one of the following is not entropy driven process. So base stacking is enthalpy driven process. So the answer here will be option A. But in your answer key it is given as base pairing, and when I referred I found that base pairing is an entropy driven process and it may be a printing mistake in the answer key. So they have asked here which is not an entropy driven process. So it is base stacking, it is enthalpy driven process. Then next one, Anjali is selling the 3 brands of dress material X, Y and Z. The production of the 3 types of material in thousands for the period 1995 to 2000 has been expressed in the following bar graph. So this is the bar graph. Find the total production of Z material in 1997-1998 is what percentage of the total production of X material in 1995 and 1996. So Z material in 1997 and 98 you will find it is around 60. So 60 plus 60 divided by now X material in 1995 and 96. So this is X, that is 50, and 96 it is 40. So 50 plus 40 into 100, that will give you 133.33%. So the answer here will be option B. Then next one. You have subjected a mixture of glutamic acid PI 3.2 and arginine PI 10.8 and valine PI 6 to electrophoresis. Which amino acids migrate towards the anode when the electrophoresis is carried out at pH 7.1? So here you should know 2 things. One is that if pH is greater than PI, it will be the protein will or the amino acid will be negatively charged. And anode is positively charged. So negatively charged amino acid migrate to the anode. So which all amino acids out of the given ones will be negatively charged? So they have told that I already told that pH is greater than PI, the amino acid will be negatively charged. So in which all amino acid pH is greater than PI, it is glutamic acid and valine. Glutamic acid and valine, the PI is less than pH. So the answer here, they will migrate towards the anode. So answer here is valine and glutamic acid. Answer is option D. Then SAM tools are used. So SAM tools is a set of programs for interacting with high throughput sequencing data. So they are used for processing aligned reads. Answer is option C. Then coming to part B. Zinc finger motif is a specific characteristic of proteins. Which statement are true about it? It is only found in eukaryotic organisms. It is found in prokaryotes also. The first prokaryotic zinc finger domain found in the transcription regulator ROS was identified in Agrobacterium tumefaciens. So, first option is not correct. It is false. Then, it is generally between 23 and 30 amino acid long. That is correct. Proteins contain zinc finger motif act as DNA binding proteins. That is also correct. Zn2 positive ion of the motif interact directly with phosphate of DNA. Zinc does not interact itself with DNA and it is involved in stabilizing the zinc finger. So 4 is also wrong. So the correct options are 2 and 3. So answer is B. Then next one. Chromatin immunoprecipitation assay is used to investigate the interaction between proteins and DNA, which is not correct statement for it. It may provide quantitative data. That's correct. It is used for high throughput screening. No, it is not used for high throughput screening. High throughput screening is used in drug discovery process and chip is used in high throughput sequencing. So option B is not a correct statement. It analyzes DNA protein interaction in living cells. Yes, in vivo also it can be carried out. It has ability to profile a promoter for different proteins. That is also correct. The next one. Deubiquitinating enzymes play several roles in the ubiquitin pathway except recycling of ubiquitin. Yes. Processing of ubiquitin precursors. That's also correct. Editing or rescue of ubiquitin conjugates. That is also correct. Assembly of unanchored ubiquitin oligomers. That is not correct. Deubiquitinating enzymes cleave ubiquitin from the proteins. So they are not involved in assembly of the unanchored ubiquitin oligomers. Answer is 30 D. The next one. Align the given types of post-translational modifications of proteins in group 1 with the participating amino acid in group 2. So N-linked glycosylation is arginine. C-linked glycosylation is for tryptophan. Phosphoglycosylation is for serine. And N-acetylation is for lysine, D2. So the answer here is option D. The next one. The additional roles of amino acid in plants besides acting as building blocks include serve to transport nitrogen from source to sink tissues. That is found in many plants where amino acids play a role. Then serve as precursors to phytohormones. Yes, certain amino acid like tryptophan, it is a precursor for auxin. Then contribute to animal nutrition? No. Participate in lipid synthesis? No. So option B is the answer, 1 and 2. Which of the following cell type in the brain are capable of phagocytosis as part of immune function? So it is the microglia. Answer is option C. Then match the following enzymes in part A with their substrates in part B. Aminotransferase. Aminotransferase is 2-ketoisovalerate. SAM synthetase. SAM synthetase is methionine. S-adenosyl methionine synthetase. So the substrate will be methionine. 2,1. Aspartate kinase. Aspartate kinase is aspartate. Threonine synthase. Threonine synthase is homoserine 4-phosphate. So the answer here will be option C. Then next one. Alternate oxidase pathway of mitochondrial electron transport chain has the following features. The correct statements are. So the alternative oxidase provides an alternate route for electrons passing through the electron transport chain to reduce oxygen. So they transfer electrons from ubiquinol to oxygen. That's correct. 5 ATP are synthesized in this pathway. Actually, low ATP is produced. Very low ATP is synthesized in this pathway. So 2 is not correct. It is a multi-enzyme complex. No. It has a molecular mass of 32 kDa. That is correct. So 1 and 4 are correct. Answer is option D. Then next one. Glutathione is a. So glutathione, it is an antioxidant found in plants, animals, fungi, some bacteria and archaea. It is a peptide. It is composed of 3 amino acids, that is glycine, cysteine and glutamic acid. So glutathione is a tripeptide. Answer is option C. Then next one. In an enzyme substrate reaction, the addition of an inhibitor did not change the Vmax value. Consider the following reaction follows Michaelis-Menten mechanism. The inhibitor is A. So there is no change in the Vmax. So that means it is a competitive inhibitor. Answer is option A. Which of the following method can be used for global gene expression profiling? So it is DNA microarrays. So DNA microarray, it is a collection of microscopic DNA spots attached to a solid surface. It is used to measure expression of a large number of genes simultaneously or to genotype multiple regions of a genome. So the answer here is option C, that is DNA microarray. Then tetracycline inhibits. So tetracycline specifically inhibits the 30S ribosomal subunit, and thereby it will hinder with the binding of the amino acyl tRNA to the acceptor site on the mRNA ribosome complex. So the answer here is option D, that is binding of amino acyl tRNA to ribosome. So the answer here is option D. The next one. The nerve centers in the brain which control body temperature urge for the eating is. So it is hypothalamus. Answer is option A. Then next one. The active cyclin CDK complex regulates the cell cycle through. So CDKs are kinases, that is enzyme that phosphorylate specific target proteins. So CDKs are kinases, so it is through phosphorylation of specific proteins. So answer is option A. Then which of the following is not an actual cell cycle checkpoint? G1 checkpoint, M checkpoint, spindle checkpoint, S checkpoint. S checkpoint is not an actual cell cycle checkpoint. Then which of the following inhibits inflammation? So it is protein C which inhibits inflammation. Answer is option C. The cell line used for the production of polio vaccine was? It was primate kidney cell line. Answer is option C. Then name the organ in which malarial parasite could reside and stay dormant for up to 1 year during the transmission of the disease in humans. So it is liver. Answer is option B. Black water fever is a special manifestation of malaria caused by Plasmodium falciparum. Answer is option A. The most active stage of feeding in lepidopteran insects is the larval stage. B. Identify the false statement with reference to Spodoptera frugiperda. So it is commonly called fall armyworm. That's correct. It invaded India in 2018. Yes, it is monophagous. It is not monophagous, it is polyphagous. Monophagous are defined as those feeding on one or more plant species within a genus. That is called as monophagous. So, but here this particular Spodoptera is monophagous. Is polyphagous. So, answer here is option C. They have asked which is the false statement. Then next one. We have to do match the following. Melanization 1 phenol oxidase. Prophenol oxidase is zymogen. Plasmatocyst phagocytosis, melanin immune response. So the answer here is option D. Then Chow-Fasman method is known to predict. So it is known to predict secondary structure of a protein. Alpha sheets, beta barrels, beta turns, alpha helix, beta sheets. All these can be predicted by Chow-Fasman method. Which of the following is a distance-based phylogenetic analysis? So there are three methods for constructing phylogenetic trees, that is distance-based methods by neighbor joining, parsimony-based methods, that is by maximum parsimony, character-based method, that is maximum likelihood, or Bayesian Bayesian inference. So here they have asked for distance-based phylogenetic method, so it is neighbor joining. Answer is option B. Then, in neurons, because the resting potential is dash, depolarization means dash while hyperpolarization means dash. So, resting potential is negative. This means the interior of the cell is negatively charged compared to the outside. And hyperpolarization happens when membrane potential become more negative. That is, opening of channels that let positive ions flow out of the cell cause hyperpolarization. And depolarization is when membrane potential become more positive. So hyperpolarization, it is more negative and depolarization is more positive. So depolarization, it happens when the channels open that let positive ions flow into the cell. So that is when depolarization happens. So the answer here will be option A, that is negative. Depolarization is more positive and hyperpolarization is more negative. So the answer here is option A. Then next one. ProCyte is used to find biologically meaningful patterns or profiles of protein. Answer is option C. Reciprocal best blast hit method can be used to identify. So it can be used to identify orthologous relations. A. Then which of the following algorithm are used for sequence alignment? Needleman, Wunsch, and Smith and Waterman are used for sequence alignments 1 and 2. Answer is option A. Needleman, Wunsch is for global alignment and Smith is for local alignment. Then next one. Which of the following is not applicable to DNA topoisomerase? Not applicable, they have asked. It has endonuclease activity. Yes. It may have an ATP binding domain. Yes. The active site of tyrosine is phosphorylated during its association with DNA. That is not correct. So option C is incorrect. Then next one. In passive agglutination test. In passive agglutination, antigen is coated on the inert material. Answer is option B. Which of the following is not the DNA polymorphism? So what is DNA polymorphism? It is presence of two or more variants form of a specific DNA sequence in different individuals in a population. So the most common type of variation is single nucleotide variant or single nucleotide polymorphism. Then you will have RFLPs, then you have variable number of tandem repeats, VNTRs, microsatellites. All these are DNA polymorphisms. So DNA methylation is not a DNA polymorphism. Answer is option B. The basic unit for Baltimore system of virus classification is mRNA formation. Answer is option B. Then which of the following cannot be used in analyzing the structure of RNA? So it is DNase 1 cannot be used for analyzing structure of RNA. Which of the following virus infects only human? Chikungunya, HIV, Zika, measles. Measles infect only humans. So answer is option A. So that's it. Thank you for watching my channel. Please do like, share and subscribe. I have uploaded previous year's PhD Biotechnology entrance exam question papers also. So you can refer to my channel for those question papers. Thank you.